Good day. This is the Professor for drwillphd, PhD. And today's Thursday, December 1st, 2022. It is 6:34 and 34 seconds a.m. Eastern Standard Time. And today's research, pancreatic cancer. So let's hop to it here. The new genetic culprit suspected in the onset of pancreatic cancer. Date November 30th, 2022. Source, Virginia Commonwealth University. The new research points to the inactivation of a previously unidentified gene as a likely culprit in the development of pancreatic cancer. The findings could alter the scientific understanding of this deadly disease and inform the establishment of novel treatments. The findings hold implications for the targeted treatment of pancreatic ductal adenocarcinoma, which accounts for the vast majority of all pancreatic tumors and is fourth leading cause of cancer-related deaths worldwide. Most patients are diagnosed at an advanced stage when the disease is already inoperable and there are no effective therapies. Extensive research has demonstrated that mutations in the KRAS gene play an enormous role in the formation and growth of pancreatic cancer. Approximately 85-90% of all pancreatic tumors have a KRAS mutation. Given its high incidence at very early stages of the disease, mutational activation of KRAS has been hypothesized as a key genetic driver for pancreatic cancer. However, there is a substantial fraction, around 10 to 15%, of pancreatic ductal adenocarcinoma tumors that harbors an offset by the conventional KRAS mutation, characterized as wild-type KRAS. This suggests that in many cases, alternate genetic drivers are responsible for the development of cancer. The new research suggests that the inactivation of NF1, a gene known as neurofibrobin 1, that holds natural tumor suppressing functions, could be instrumental in the onset of pancreatic cancer, either in tandem with KRAS, bolstering its cancer-driving properties, or even before any mutations occur in the KRAS gene, in partnership with TP53, the most inactivated tumor suppressor gene in human malignancies. The researchers determined that removing neurofibrobin 1 in mice without KRAS mutations directly resulted in the early development stages of pancreatic tumors, but also enhanced the cancer-driving function of KRAS in mice with mutations. Researchers found that genetic inactivation of neurofibrobin 1 dramatically accelerates the formation and progression of KRAS-mediated pancreatic cancer. And this study raises the proactive possibility that targeting neurofibrobin 1 and mutant KRAS bearing pancreatic tumors might create vulnerabilities that can be exploited for therapeutic advantage. Additionally, the researchers observed a strong association between neurofibrobin 1 and P53, another protein widely known for its tumor-suppressing functions. They found that the simultaneous inactivation of both neurofibrobin 1 and P53 directly correlated to pancreatic cancer growth, regardless of any mutations in the KRAS gene. The notion that the combined inactivation of neurofibrobin 1 and P53 represents the alternative initiating event in the pancreatic ductal adenocarcinoma provides an unprecedented platform for the future identification of novel targeted treatment options for pancreatic cancer. And this is the Professor of Microbiology, Bioinformatics, and Pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.